Hi everyone, welcome to Chapter 6. This is the unifying chapter for this unit, Unit 2, which is about cell signaling and communication. I've put cell communication here, but the truth is it would be communication, integration, and or response. This unit includes this general chapter, and then we get into individual systems, like for instance endocrine in Chapter 7, neurological in Chapter 8, then Chapter 10 is the senses, and Chapter 11 is the autonomic system. So we have 5 chapters in this unit. Unfortunately, it's because of figures like this that cell signaling, signal transduction, is a scary topic for many. Very difficult to penetrate when you have a figure like this, which, by the way, is apoptosis. We talk about apoptosis being regulated and controlled cell death. Well, this is what is generally known. This is some of the more commonly known areas that regulate aspects of apoptosis. If you look at the bottom here, you can see that when we get here, we have DNA fragmentation and so forth. That's one of the final steps in this effect of inducing apoptosis in the cells. But as complicated and as scary as this figure might seem, especially to somebody who hasn't had a whole lot of biology, it comes back to, and it looks like one of these 6-ball juggler kind of things, just so many things going on, it's hard to follow an individual ball. But the truth is, what we're dealing with here is really just a set of single type ball throws. This interaction from here to here would just be one ball, but it would be similar to this interaction from here to here. Again, a similar situation. All signaling, no matter how complicated and or nuanced it might be, fundamentally follows this pathway. You have some kind of a signal. You have something that can perceive that signal. You change the shape of this receptor and that triggers a message either inside the cell or having to do with the cell, leading to a response. So we have a receptor, that's the perception side. We change the nature of the signal. So maybe this is a chemical message out here, and these messages here are physical, like for instance in a cytoskeletal receptor, leading to appropriate response, but I would put in parentheses an S there, responses. And that's why it gets complicated. It's a bunch of simple processes occurring simultaneously, like a 3-ring circus. But it always comes back to, if we look at this figure again, this fundamental pathway. And let's just look at this. We have the signal up here. It might be a chemical message or a physical one. It goes through, that would be up here. So here's our receptor. Then we go through a signal transduction pathway going somewhere. Like for instance, let's do this one. Let's go down here and let's go to the mitochondria. One of the things that happens in mitochondria is you release some of the contents and then that triggers further responses. And so there's a simple one. We go through here, maybe you're this way, that sort of a thing, leading to mitochondrial release. Just like that. Or maybe we have another figure that goes like down here to DNA fragmentation of some kind. Again, it's the same pathway. It's just that it's going through some different sets of proteins. It's just that simple. And once you get that, then a figure like this is really just, looking at the pieces. This is actually a reference figure. Very few people have this memorized. They just look at it to make sure they've covered all their bases when they're doing research or something. But it always comes back to this basic reflex pathway that we literally saw in Chapter 1. Some sort of a way of perceiving a change or a signal of some kind, some way of linking that perception to the appropriate responders to get the appropriate effects. Negative feedback, positive feedback, or feed forward. The majority of cell-to-cell communication paradigms, and there are multiple forms of this, the majority, though, however, fall into 3 major groups. We have electrical communication. And this involves changes in the membrane potential, usually associated with the changes in permeability of the membrane to ions. We have chemical signals, and this is by far, this dwarfs everything else. This is by far the biggest category, where we have a diffusible signal that's secreted into the extracellular fluid and moves to either the same cell that secreted it or to others. And those others must have a receptor for that signal if they are to respond. So chemical signals. And then mechanical signaling. This is often through some of those cell adhesion molecules that we talked about, those CAMs, and they are usually connected to the cytoskeleton in many cases. We are going to talk about contact-dependent signaling, but I would lump that into more chemical side of the equation rather than mechanical. In any case, though, the target cell, the cell that is meant, the signal is meant to induce a response in, must have that receptor, or the response will go lacking. So we must have a receptor, whether that receptor is intentional or accidental. We have receptors that were never meant to be receptors. It's just that happenstance made them receptors in a given circumstance. Let me give you an example. The thermoreceptors on your tongue for heat also happen to be chemoreceptors for a molecule known as capsaicin. So if you go to your favorite, you have your favorite hot sauce, favorite salsa, what happens? You take a bite of that hot sauce or that salsa and it feels like the temperature in your mouth goes up. But we used to do an experiment in 165 where we would measure mouth temperature before and after differing heats of sauce, and we found no change in the actual temperature of the mouth, even with very, very hot sauce. It was all chemically gated. Nevertheless, those receptors are also temperature gated, so it's an accidental receptor in some ways. So what I'm going to do now is give you some examples of these types of signaling. For instance, electrical signaling, a little bit about chemical signaling, because there are some different paradigms here because this is such an extensive category, and then mechanical signaling. So we're just going to give you a few examples or one or two examples of each. Electrical signaling fundamentally involves membrane permeability and the ability of ions to, first of all, be differentially distributed across the membrane. So this picture down here is showing the sodium-potassium ATPase, pushing potassium in, pushing sodium out from unit 1. Then we can have channels in here that then allow those molecules to move, those ions to move, and that results in an electrical field across the membrane. If we change the permeability to ions, we change that electrical field, and that can be the basis for signaling. With chemical, like I said, it's a big category. We can talk about different kinds of chemical signaling. But the commonality across all of these is that the signal is a molecule of some kind or an ion in some cases, but it is diffusible. That's the main criterion here. For instance, you can have autocrine signaling where a cell secretes a molecule or a signal of some kind that then binds and interacts with receptors on that same cell. Beta cells in the pancreas, the ones that release insulin, have insulin receptors. And that's meant to control how much insulin is released so you're not releasing too much. We have an intracellular type situation. This is where a second message of some kind or an intracellular signal arises, and it's able to diffuse through those gap junctions and lead to targets and effectors in the target cells. The one that comes to mind most often is calcium. So we have calcium ions that are released, and they can diffuse across and then affect calcium-sensitive proteins in the next cell. Like in, for instance, smooth muscle and cardiac muscle both use that mechanism. Similar to gap junctions, only this time we are diffusing extracellularly. This would be called paracrine. There's even a phrase called juxtacrine, where you have the cell adjacent to the original secreting cell. We're only talking about maybe 5 cell diameters max here. Very, very localized phenomenon. Paracrine, just like paracellular is transport, paracrine is a local signaling phenomenon. And then you have the true long distant signaling, and this can be, let's say, between more than 5 to 10 cell diameters. Oftentimes, endocrine signals involve more than one extracellular compartment. It's seldom or never that you see these molecules getting into the blood. But in endocrine, molecules first going into the extracellular space and then the blood is quite common. So distance signaling falls under the endocrine, so more than, say, 10 cell diameters. In the mechanical side, like I said, most of the time what we're dealing with is some aspect of the cytoskeleton. Maybe at the beginning you have a receptor and it causes a conformational change in the cytoskeleton that leads to some kind of metabolic message that then affects response. In other cases, you may have a mechanical signaling that goes almost all the way to the nucleus before affecting a response. Either way, what's going on? The signal is force in nature, is in its mode. It's mechanical force or shear or stress or tension. That's partly why you can get differential responses to different types of exercise in your muscle. You can do pull-ups, and you can do lots of pull-ups, you can get strong, but you can also change the nature of your muscle by doing negative pull-ups, where you're doing negative exercises, where you're lowering yourself down slowly. And that will lead to differential responses in the muscle. The muscle will respond not only in the immediate sense, but also in the slow genetic sense. But this is mechanical signaling. But it's always these fundamental ingredients. Remember, though, that the receptor does not necessarily have to be on the cell surface. It doesn't have to be in the plasma membrane. It could be inside the cytosol. It could be all the way down in the deepest of organelles, like in the nucleus. The general rule of thumb, again, there's always exceptions. General rule of thumb is that the deeper the receptor is, the more likely it's going to affect genetic changes in the cell. The deeper the receptor is, probably the more lipophilic the message, the first message is in that. And we'll talk about that when we get to chapter 7. The more exterior the receptor is located, the more likely you're going to include a fast cytosolic response. Fast cytosolic response could be exclusively cytosolic and you're getting little or no genetic influence. Or it could be a fast cytosolic response on the way to a slow genomic response. Most cells respond in multiple ways to a given stimulus. And so you're often seeing both of these things here. But if you were to hedge your bets, the further to the exterior the receptor is, the more likely you're talking about a fast. The more interior the receptor, the more likely you're going to see a predominant response being genetic. Same story, just another picture. This one's from your book. Again, just that same mode of reception, some sort of a relay where we change the mode of the signal. For instance, let's say this was light. So maybe we have a receptor here and we cause some sort of a second biochemical message. It's no longer light. That leads to some kind of a response in the cell. But it's the same motif, regardless of what the mode of the stimulus is, you have to have a receptor. You're going to have some connection between the receptor and the response. Sometimes it's one step or almost no steps. Sometimes it might be many. Now, contact-dependent signaling. You could certainly make the case that the cytoskeletal type elements that we talked about with mechanoreceptors are contact-dependent. Nevertheless, there are more commonly, what you see is that there's a molecule that's put on the surface of one cell that interacts with a receptor on another. And this resembles the pathways that look like diffusional, chemically gated signals. You're not affecting the cytoskeleton so much, you're causing second message and metabolic changes in the cytosol to occur. And this is certainly extremely common in the immune system in our bodies, that different members of the immune system talk to each other in a sort of a quasi-chemical way through these contact-dependent signals. Just reiterating here, autocrine, Adjacent, would be paracrine, and maybe a couple of cell, 3-cell diameter, 5-cell diameters would all be in this paradigm. Autocrine's the same cell. Paracrines are adjacent cells, usually up to between 5 and 10 cell diameters. And then we have the true long-distance diffusible signal that we call the endocrine signal, classically known as hormones, but the lines have blurred and now we see a spectrum between very localized phenomenon and systemic phenomena. What we have going on in these situations is that there's a signal that's released from a cell. It diffuses from the cell into the extracellular compartments, including the bloodstream, and then diffuses out through the system, occasionally leaving the bloodstream. If it leaves the bloodstream and interacts with a cell that doesn't have a receptor, you're not going to get a response. That may be because it doesn't have a receptor, was never meant to respond to that signal, or it lacks a receptor or that sort of a thing. But cells that do have a receptor, depending on the receptor and the second message, will drive the response. Let me give you an example of what I'm talking about here. That phenomenon of the same signal leading to different responses in different types of cells is called a pleiotropic response. A pleiotropic response. We'll see that when we get to chapter 7. But for instance, the active ingredient in the drug formula for Rogaine diffuses through the, is applied, and it acts on receptors in the skin to trigger vasodilation. And so the blood vessels dilate in the areas that are exposed to this drug, and that promotes hair growth. On the other hand, the same active ingredient, if administered a different way, will trigger vasoconstriction in the reproductive areas, and we call that drug Viagra. They are very, very similar, if not identical, types of signals. It's just that one's mediating a vasoconstriction response, and the other one's mediating a vasodilation response. Hormones, again, were the classic signal, but we're going to talk about some other signals that also behave like hormones. So long distance communication, fundamentally further than 5 to 10 cell diameters. We can see this, like I said, with electrical signaling and in neurons, and we can see hormone signaling. We'll talk about these when we get to chapter 7. But we're also going to, in a few slides, talk about a newer category of molecules that behave in some ways like hormones called cytokines. Most of these long-distance communication pathways are not monolithic. That is to say, they are not exclusively electrical, they are not exclusively chemical, often involve multiple steps. Like for instance, in this one, we have an electrical signal that can travel as much as a meter or more, and then it gets to what we call a presynaptic terminal that releases a diffusible signal that leads to a target cell, like for instance, a muscle fiber or a gland, causing it to respond. Again, here's that electrical signal. We have the chemical signal here at the synapse. I would categorize that as paracrine. Some people would say neurotransmitter. It's just the reason they say that is because the kinetics are really, really fast here. We're talking about millisecond kinetics. It is still a paracrine phenomenon because the molecules are diffusing and encountering a receptor. But that's not the end of the story. Not all of that chemical signaling in neurons is short distance. For instance, in your posterior pituitary, and we've already talked about this with oxytocin, you have neurons that originate a signal, but rather than hooking up with either a muscle or a gland, that presynaptic terminal actually releases molecules into the extracellular space in the bloodstream, leading to distant targets. So for instance, the neuron secretes oxytocin. It diffuses through the blood, doesn't react with, let's say, your quadriceps muscles all that much, but when it hits that uterine smooth muscle, it triggers contraction. So again, we call these neurohormones, but it's the same story. It's just that the signal originates from a neuron rather than from a specialized endocrine, non-neuronal endocrine cell. Now let's get to cytokines. And the phrase that I say is cytokines blur the lines. That's because cytokines have ways of acting like local signals as well as long distance. They can act in long distance ways. Let me give you an example. Anybody who's experienced allergies is experiencing a long distance systemic type of a signal. Itchy eyes, watery nose, that sort of a thing. Those are long distance, like hormone-like signals that are mediated by a cytokine known as histamine. On the other hand, if you get a mosquito bite on your skin, you very seldomly get itchy eyes and runny nose and that sort of a thing. But you do get a little welt right where the mosquito bit you. That's also mediated by histamine. You know how you know? It's because histamine mediates the itch that you get in one of those mosquito bites. But it's acting locally in that situation, and it's acting in long distance in the allergy, especially severe allergy situation. The other big difference with cytokines from, say, hormones or neurohormones is that all nucleated cells, to our knowledge, can synthesize and secrete cytokines. A classic example of this would be interferon. When a cell is infected by a virus, one of the most common cytokines that is released from that cell, and it's shown right, it's just the molecule, I see it right here in the figure, is interferon. This just happens to be interferon gamma. Interferons will diffuse away from that cell and trigger other cells, like for instance, let's say this guy is secreting interferon, trigger other cells to say, hey, there's a virus around, prepare yourself. And those cells will begin manufacturing antiviral proteins and molecules to prevent virus from infecting those uninfected cells. So it's a classic example of a nucleated cell releasing a cytokine. In development and differentiation in utero and even sometimes postnatal, cytokines are involved in development and differentiation of tissues, appropriate neural patterning in some cases, appropriate growth and or cell division in some cases. So they work both as autocrine and paracrine. But most of the time that I'm familiar with is involved in the organ patterning and specific differentiation of tissues. We see cytokines in the bone marrow affecting the development of red blood cells, or those same precursors that would have normally gone into red blood cells are now shunted over into white blood cells, cytokines. Cytokines, like I said, in allergies, stress and inflammation, some cytokines will act on distant targets. Histamine is a classic example of this. Bringing it back though, whether it's cytokines or hormones, neurological, electrical signals, and so forth, it's always that same basic paradigm. Probably the best advice I can give you is to try to apply this paradigm to whatever pathway you're looking at. Right? It's an input, there's some kind of integration, sometimes amplification in some cases, I'll show you what I mean, leading to effectors activated and responses. And just another picture of the same thing, though I've added a little more detail to this one. We are going to transition into that second message here in a little bit. The possible responses, changes in protein function, like for instance myosin in a muscle contraction, or transcription factors that change the effect of genes, which might affect, say, protein expression or cell growth. Certainly movement, if myosin would be in the movement category, or perhaps inducing apoptosis, like in that scary picture I showed you earlier, or some other metabolic activity, like secretion. It's always this same story. What we're going to do now is move into second message systems. We're going to move to this transducer intracellular signaling molecule level. One common motif is what we would call a cascade of kinases, a protein kinase cascade. So we have a receptor that's activated, and when that receptor is activated, it changes its shape ever so subtly, that it can now lead to interaction and activation of some protein. Well, in this model of second message, that protein happens to be a kinase. And that kinase will then start phosphorylating targets, and it might phosphorylate a few molecules or it might phosphorylate hundreds. So we have one receptor that now has hundreds of second message. And guess what? That message here is also a kinase. And if each molecule of this is going to phosphorylate hundreds of these molecules. So now we've got an amplification in the thousands or tens of thousands or hundreds of thousands or millions. So a sequential activation of kinases that can lead to amplification or certainly bifurcation of the pathway to multiple responses, not just genetic and not just cytosolic. But at some point you're going to phosphorylate a transcription factor of some kind, and it's going to bind the DNA and change the gene expression in that cell. Another very, very common cleavage cascade, notable in blood clotting, notable in apoptosis as well, is this what we call proteolytic cleavage cascade. So we have cleavage, which is chopping up a protein selectively, usually. Talking about selective cleavage of specific locations in a peptide. So we activate the receptor, and that turns on enzyme 1, which will cleave a molecule or so of that. And when it cleaves it, now we have an active enzyme for number 2. And this one now will cleave maybe 100 molecules of the next substrate, which is an enzyme number 3, which is also a protease that then cleaves 100 molecules for every one that's created, and now we have 10,000, and so forth and so forth. Well, it's an activation of a set of proteases that selectively cleave a target of some kind, and again, amplification and bifurcation of response in some cases. So here's just a representative example of what we would call the more traditional second message. The more traditional second message is either an ion or a metabolite of a molecule, like for instance, a metabolite of ATP that we call cyclic AMP, or a metabolite of GTP that we call cyclic GMP, or release of calcium and so forth. These are generally intracellular diffusible signals that act as cofactors on other proteins. And so here's our receptor. It's a G protein coupled receptor, and we now have an active G protein that goes and turns on an enzyme in the membrane. That enzyme starts metabolizing a molecule. Maybe it's ATP, and now we are making cyclic AMP. If you ever heard the phrase adenylyl cyclase, and I'm not going to test you on that, but adenylyl cyclase is the enzyme that converts ATP to cyclic AMP. And you're going to make thousands and thousands of those molecules, again, amplification and potential diffusion and bifurcation of the second message into other pathways, leading to effectors, leading to responses. This is the classic second message system. The other two aren't necessarily classic, but they are common. Putting these things in place, I've got a little bit of an animation here. Here we have a diffusible signal outside the cell, binding a cell surface receptor. If this molecule cannot penetrate the plasma membrane on its own, there is no way that it's going to bind an intracellular, much less a nuclear receptor. There are very few examples of non-permeable molecules getting through because of a transporter or something. Drugs, that may be the case, but endogenous signals, not likely. So if it's not able to pass through the plasma membrane, it has to bind a cell surface receptor. We get some kind of a rapid cytosolic response that may or may not lead to a genetic response. Almost always when you have a cell surface receptor like this, you're going to get a rapid cellular response, and you can bet money on that. Some examples of rapid cellular responses include changes in metabolic function, activation and inactivation of enzymes. Again, the classic example is muscular contraction. Acetylcholine is released from a neuron. It binds an acetylcholine receptor, leads to calcium release. That calcium release causes a rapid association of myosin and actin for contraction. Vesicular transport's another good example of this, where you are triggering a signal. that leads to the exocytosis of a signal of another hormone, or perhaps we talk about receptor-mediated endocytosis. That's where a molecule like a nutrient will bind a receptor, and that will cause the plasma membrane to bend in, and you phagocytose those molecules. Movement, we already mentioned muscle. I won't do that again. When it comes to intracellular receptors, though, you have to have a diffusible signal of some kind. It has to be lipophilic if it's going to make it through into either the cytosol, or very lipophilic in the case of what we call a nuclear receptor. So we have a cytosolic receptor and a nuclear receptor. Turns out there's also some in-betweens, like for instance, the estrogen receptor binds estrogen in the cytosol, but then moves to the nucleus to mediate changes in genetic expression. Again, the further in, the more likely you're seeing a genetic type of, a slower type of response. Cytosolic and nuclear receptors. On the surface of the membrane, though, the receptors are more varied. So if a cytosolic receptor and nuclear receptors are more likely to be transcription factors, then plasma membrane receptors are going to have a much different configuration, because they often will mediate rapid cytosolic events. So we've got four major groups here, and again, we're going to break out the individuals, give you some examples of each. Receptor channels, sometimes known, and you're going to hear this phrase again this semester, as ionotropic receptors. Ionotropic. We have receptor enzymes. Adenyl cyclase, like I mentioned in the previous slide, is a sort of an example of that. We have this really fun one, albeit often a little more unfamiliar to folks who haven't had biology for very much. This is called a G protein coupled receptor. It's got 7 transmembrane domains. You can spot it. But it associates with a second message type mediator, a second message protein called a G protein that interacts with either GTP or GDP. It can switch from active and inactive states depending on whether it's bound to GTP or GDP. To illustrate why I'm talking about this, even though it's not familiar to many of us, 2/3 of the drugs in the known pharmacopoeia, that is to say, the lexicon, the dictionary of all the pharmacoactive compounds, 2/3 of them affect G protein coupled receptors. And then lastly over here, the one I'm going to talk about the least, we've sort of alluded to them, like in the case of cadherins. This family is known as integrins because, like cadherins, they will help attach the cytoskeleton to extracellular matrix, but they are also gated for the presence or absence of sometimes chemical signals or other types of contact-dependent signaling, and they often move through cytoskeletal mechanoreceptor kinds of situations. Going to start with those receptor channels, those receptor channels, like I said, ionotropic. In this case, we have a chemical message. So without the chemical message, that receptor is closed. But when that chemical message binds, it triggers a conformation change, and now the channel opens, and we have altered the permeability for an ion of some kind, or maybe water. I haven't ever heard of a gated channel for water, not to my knowledge, but certainly ions are gated channels. And so you've changed the permeability based on whether you're opening or closing this channel. And that can lead to changes in the electrical voltage or other types of second messaging. Now this receptor channel is not gated to the outside. It is actually gated to a molecule on the inside. So some of these channels have receptor domains on the outside of the cell. Others will have receptor domains to second message from the inside of the cell. That may be something like cyclic AMP or cyclic GMP. Again, another intracellular gated channel. But they're always gated to the presence of absence of a chemical or a molecule. Most of the time we're talking a diffusible molecule. With rare exception, there may be a contact-dependent type situation. Receptor enzymes, we're talking about what happens when the receptor is activated, it can facilitate metabolism of other molecules. Here's one where we have a receptor that's activated, and when it's activated, it starts converting GTP into a second message, cyclic GMP. So this receptor has guanylyl cyclase activity when it's activated. It's turning the linear GTP into a cyclic GMP. Most common type of receptor enzymes are actually kinases. That is to say, when the receptor is activated, you have a kinase domain that now allows for phosphorylation of targets, post-translational modification of targets. So I'm going to show you some animations here in just a second, and hopefully they work for you. A common situation with these kinase type receptors is that they'll bind the ligand, they'll bind the first message, and they will dimerize. They will associate with each other, and when they do, the one on the left will phosphorylate the one on the right, and the one on the right will phosphorylate the one on the left using ATP. Think of it this way. If this is a person with their arms folded across in front of them, they're not really ready to accept a hug, right? They're not really ready for interaction. But once those arms get spread out, like in the case of phosphorylation, now we've rolled out the red carpet, or we've spread our arms wide and are ready for interaction. That's what we mean by activation. We phosphorylate the target, it changes its shape, and now it can interact with other proteins, leading to second message. And so here's an animation. Hopefully it's working for you. If it's not, what I'm describing here is that you have some kind of a growth factor molecule, and it binds the receptors individually, and then we get a dimerization effect. This is actually a tyrosine kinase receptor, which means that the targets on the opposite protein are the side chain tyrosine. So those get phosphorylated, and that leads to changes in activity. And so here we go. Those intracellular domains cross. They get phosphorylated by the kinase domain in the reciprocal protein. And now we have phosphorylated residues that will allow binding of relay proteins that lead to subsequent signaling inside the cell. In this case right here, just for example, this GRAB2 sauce binds that activated receptor and actually will phosphorylate targets in a kinase cascade. So again, this interaction of proteins that facilitates the creation of a second message. Our next category of receptors are the G protein coupled receptors. It used to be called serpentine receptors because of the 7 transmembrane domain. Again, very druggable target. Why they're called G protein coupled receptors is because of the nature of the second message. The second message is an activated G protein. When there is no ligand, or they say here agonist bound, the receptor protein will hold this trimer, it's an alpha, a beta, and a gamma, this heterotrimer in an inactive state conjugated to GDP. But as soon as that agonist, that ligand binds, changes the shape of this a little bit, this dissociates along with the regulatory subunits. We swap out GDP for GTP, and now this alpha can go modulate the activity of downstream targets. And that's what we're showing here. So in an inactive state, prior to the binding of ligand, we have this trimer, but as soon as the ligand binds, that alpha is released and it swaps out the GDP for GTP, and then now it will lead to second message interactions. This one is neat because it leads to a signal, but then it can self-regulate. At some point that GTP will be hydrolyzed back into GDP, and this thing will shut down. So it's sort of like a time-dependent type switch. You turn it on, and then a timer sets, and then it'll be active for a little while, and then it'll hydrolyze this GTP and go back to this inactive state. It's really kind of an interesting mechanism once you get to know it. Integrins, like I said, are often associated with extracellular matrix like cadherins, and they're often involved with cadherins in making sure that the cell knows that it's in the right place. It can be a mechanically gated type situation. It can also lead to second message that's chemical and biochemical in nature. But the point here is that they are embedded in the membranes, and they serve an anchoring function as well as a signaling function to the cell. One of the most common things is to tell the cell to not die. Here's an integrin pathway. Again, the idea here's the receptor and the ligand. Well, here we have the transduction events leading to effector proteins that may involve genetic or fast cytosolic responses like proliferation, survival, and migration. These next few slides actually make me the most nervous of all the slides in this presentation. And the reason being is that these figures are often very complicated. You got a whole bunch of things going on. And if you're just trying to kind of keep a 10,000-foot view on it, it can be very, very difficult to internalize. So what I've done is I've blocked out the other molecules here just to show you one single circumstance. One single event in what could be a much larger pathway. Here, this is our ionotropic channel receptor. We have an extracellular signal of some kind. In this case, it's a molecule. It binds specifically on that channel, causing it to either open or close, altering permeability of ions across that membrane. We can achieve the same thing using a different pathway. Like, for instance, maybe we have a G protein coupled receptor, and when it's activated by a different signal, it can activate that G protein. That G protein goes and interacts maybe with a channel and makes a second message that then interacts with the channel, causing a change in the permeability. So we could have the same net effect with two different receptors, two different signals. That's why we have these sort of overlapping and redundant systems, is so that we can have a similar pathway responding to different signals, or perhaps we have different pathways responding to different signals. What we don't know is what ions move across here, so maybe this one's different from this one. Just depends on the circumstance. But it is an or situation. Maybe we have this, maybe we have this. That's why we want to break these out individually, because trying to digest them simultaneously can be really difficult. But let's look now. So like, for instance, here's our G protein, but instead of interacting directly, now it's creating a second message. And then that second message is what's triggering that ion channel. Again, that's a third possibility here. Ionotropic, and then G protein would be what we call metabotropic, because you're doing some kind of metabolism before you get your response. So we have ionotropic, that's a direct channel receptor. That's the most very, very rapid. But maybe we want to do it a little bit slower, or we want to be able to shut it off after a period of time. And we have the metabotropic side. We're going to see this a lot when we get into sensory perception. So that's why I'm giving you these words now. You'll see them later on. You can write them down now, and hopefully they'll sink in a little more when we talk about them later. Nevertheless, maybe that second signal was a receptor enzyme activation. So we have cyclic AMP, or we cleave some phospholipids, and those metabolites then open up calcium channels in the endoplasmic reticulum, and the list goes on and on and on. But it's the same story: signal, receptor, some kind of a message response, or maybe signal, receptor, and then the message is internalized in the structure of that protein, response. All sorts of possibilities. That's why learning the generalities gets you further than trying to learn each and every specific. Or even perhaps the ions over here on this one, or the ions here could be second message. Maybe our effectors are voltage sensitive. Like, for instance, let's say we change the permeability of this to sodium. Sodium comes flying in. Now the membrane potential is changed, and that then leads to voltage sensitive proteins now changing their behavior. Very common. We're going to see this in neurons in just a little while, where we regulate permeability. When the permeability changes, we get a change in voltage, and then the effectors are voltage sensitive, leading to some kind of response. Really cool. Keep it simple and then add layers. Then it gets easier to manage. Like this figure right here. This figure is a pain because it's so abstract. It could be this, could be that, could be the other thing. You can do this once you have a sense of the basic signaling. So we have a receptor. Maybe we have a protein kinase cascade leading to a response. Maybe we turn on an enzyme that makes a second message that leads to protein kinases, or you name it. It's trying to show you all the possibilities at once, and I believe in the intent. However, it's really difficult to try to go at this first. Second message, yeah, I just had that in probably from a previous point I made in another class. Nevertheless, for our class, I would like you to try to read pages 171, 173 carefully. Take good notes here. If you can deal on the level that the book is dealing with here, you will fly through some stuff down the line. Do this now, and you're going to make what could be really difficult something much easier, I believe. So putting the front loading that work. Start with the section titled The Most Rapid Signal Pathways, and then up to Mini Lipophilic. So everything in between those two sections, including the first part, not necessarily including the second part. So those pages for you. But it's always back to this. Start here. Try to recognize the trees inside the forest. Try to recognize the general process that's occurring, and no pathway will defeat you. It's just a matter of learning a few details here and there as needed down the line. And so that concludes chapter 6, a relatively quick chapter. We're going to have a bunch of bigger ones later on. Sorry if I went a little bit long. I'm hitting 46 minutes. It's longer than I would have hoped, but that's how it goes, and hopefully not imposing too much on you. Thanks for your time, and I will talk to you again soon. Take care.